All right. All right. Today is cross night.
Yes, sir.
It must be fruitful. What we expect to come in exam. In life science, we don't set new questions. Is it clear? And don't forget that we are going to write one paper. And that one paper is going to have everything from DNA until the end of the ear. So which part of the ear is last to be talked about?
The
ear defects. All right, let's start with the DNA, code of life. Code of life. What are you supposed to know on DNA code of life? Number one, you need to know the structure. Describe the structure of DNA. I told you that if you want to remember this is what you are supposed to do. What are you supposed to do? Draw a simple structure, and then you will be able to remember. Describe then what you see. So for example, remember that DNA has a sugar, a nitrogenous base, and a phosphate, isn't it? A phosphate, a sugar, nitrogenous base, a sugar and nitrogenous base. In between here, you have weak hydrogen bonds. You see, now you are drawing it, yes, but you are describing it. Is it clear?
Yes, sir.
Yes. And then now, decide the phosphate, you have so like that. So it continues. So based on this, you have to know how to describe. And if you know how to describe, it means that you know how to label it. So if a question comes on this, it means that you can get marks for description of DNA. Number two, you can get marks if they ask you the brain structure and ask you to label. It's rare to ask you to draw. It's very rare. They can ask you the structure of DNA on its own, or they can base on the nucleotide alone. When they ask the structure of DNA, we talk about the whole thing. When they ask the nucleotide alone, you only talk about this. Is it clear? All right, now you're going to label for me. What is this? A phosphate group. What is this? Sugar. This is a sugar, ne? Any sugar? Deoxyribose sugar. What's this? Nitrogenous base. So it depends on which kind of nitrogenous base they have put there.
Yeah.
But based on that, it can be either adenine or thymine. No, why? Because of the double bond. Well, here, guanine and cytosine, triple bond. Is it clear? Yes. So if they ask you to identify them, then it's either adenine or thymine. Or if it is triple bond, it is either guanine or cytosine. All right. How do you call this? Nucleotide. How do we call this? No, this. Only this without a base. Guys, use your phone properly. Please put it in silent. So what I'm saying, how do you call this? We call it DNA backbone. DNA backbone. Yes, it's a backbone. So if they ask you about this, that's what they're talking about. Is it clear? How do you call this? How do you call this? No hydrogen bond. No hydrogen bond. I told you it's supposed to be. How do we call these bonds? How do you call them? Weak hydrogen bond. Don't call them hydrogen bonds. The word weak must be there. If you check this book, the word weak is what? Is even changed in a different color to show you that we need to see it. Not just hydrogen bonds. Yes. How do we call this? This. How do you call it? No, not just base pair. Complementary base pairs. I see. It's just a small thing which is gonna make you to go off. Not because you don't know. We just have to need some small revision so that you boost up. Hence, that's why you are here. So this is what you call complementary base pairs. Is it clear? All right. I think we are done with this DNA. If they ask you to describe, well, what are you gonna say? DNA is double helix. Is this double helix? Is this double helix? This is not double helix.
No, it's just two strands.
Double helix. Remember, this is helical. I told you, this is helical. Another helical. You understand? Because there are two, then you call it double helical. But when you're describing, it becomes double helix. So its nature is double helix. You said double helical, 0. Double. Sean, double. DNA is double helix. Yes, with the two strands. Because those are two different things. Yes? Made up of nucleotides. With a phosphate. Yes? Oxyribose, sugar. And nitrogenous base. Is it clear? I.e. adenine, thymine, guanine, and cytosine. Mentioning them is a tick. Don't stop there. Whereby adenine goes with thymine and guanine goes with cytosine. That's another mark. Mentioning the 4 bases is a tick. You understand? And then mentioning the how do they pair is another tick. As there is no way how you can pair. Impossible. I don't think this can happen. All right. Is it clear?
Yes.
Whereby adenine goes with thymine and then guanine goes with cytosine. The bases are linked together by weak hydrogen bonds. By weak hydrogen bonds. Is it clear? So when you're describing, you just look at it and then you tell us what you see there. You will get the marks. That's DNA structure. Remind me, who discovered the structure of DNA? Who discovered the structure of DNA? That DNA is double helix with complementary base pairs. We don't have anybody called this something. We don't want those answers. Something. What is that something?
Some countries just say like yes.
What's your name? What's your surname? So why is it possible that you can remember your name and the surname, and then the others you just want to talk about only the name. You know what? You know the reason why you have to know both names, or the name and the surname, yeah? Is because they will say that James Franklin. Because when you cram the James, I say, it's this one. We don't have James Franklin. It's supposed to be James Watson. Francis Crick. James Watson and Francis Crick, they are together. So they discovered the structure of DNA. Is it clear? And then Rosalind Franklin and Morris Wilkie. They are also what? Together. Is it clear?
Yes.
So for them, they discovered using the X-ray diffraction pictures. Is it clear? Wait, just one moment. If they talk about X-ray diffraction pictures, then you talk about Morris Wilkin and Rosalind Franklin. Is it clear? Huh? Oh, can we continue now?
Yes, sir.
DNA is done. What about RNA? It's the same thing. RNA has one strand. Is it clear?
Yes.
What is the major difference between DNA and RNA? What makes it to be different and unique? It's only one thing.
DNA is deoxyribonucleic sugar and RNA.
Or
Something like that.
and
Oh,
RNA has
uracil.
instead of
Yeah, you see that uracil?
RNA.
It makes the structure to change. You understand? So when you are describing, guys, when you are describing, make sure you don't forget these points. DNA has thymine, RNA has uracil. DNA has DNA is double stranded, RNA. Please don't mix this. DNA is double helix, RNA is single stranded. Wrong. You are mixing oranges with mangoes. How can you compare them? It's like Notando and me. You ask who is beautiful? I'm a guy, you're a lady. You can't compare me with you. You understand? So when you want to compare me, you have to compare me with a guy. You understand? Yeah. So that you have mango and the mango. So when you say double helix and then you say single strand, those are two different things. So double helix is not helical. You understand? Double stranded, single stranded. You understand?
Yes.
And then uracil on RNA, thymine. I see. And then on DNA, deoxyribose sugar, ribose sugar. Those ones are simple. Yeah. Now the question comes. Give me two differences between the nucleotide of DNA and the nucleotide of RNA. The question is not asking, give the difference between DNA and RNA. Do you understand? That's how we try to confuse you and you go off. Because you are you know, when a student see the question is easy, they rush, they fail more. The question is too difficult, they spend a lot of time, they pass it. Which is wrong. When you say a question is easy, take more time so that you maximize all the marks. Okay, here. Number one, the difference between DNA nucleotide and RNA nucleotide. Two of them, I want. The double. The double. All right, Laika.
So, in the nucleotide of DNA, the ribose sugar is the sugar is deoxyribose sugar, and in the nucleotide of RNA, the sugar is
ribose.
no, I don't know. But yeah, it's ribose sugar for RNA. Yeah.
Are you sure?
Isn't it?
I'm asking.
Yes.
Are you sure?
I think so.
I don't think so.
I tried.
You know why you're doing that? Because you are not here on the day of the quiz.
Yeah. So I
Ne?
present with the bond voice.
I asked a student, are you sure? Tell me the answer. And go, are you sure?
Yes,
Which one are you going with? Which one are you going with?
Never mind.
Went with the wrong one.
That's not fair.
Okay.
That's not fair. Thank you.
No, that's the reason why some students, they write the answer in the paper, they cross it, they put the wrong one.
Yeah, so because you are, I think I shame.
No, the answer is correct.
Okay.
RNA nucleotide is made up of ribose, sugar. DNA nucleotide is made up of, they are looking for only this, and this. DNA nucleotide is made up of deoxyribose, sugar. RNA nucleotide is made up of ribose, sugar. The first difference, RNA nucleotide is made up of ribose, sugar, while DNA nucleotide is made up of deoxyribose, sugar.
Sir, can you also mention the when it comes to the differences, the weak hydrogen bond?
No, they're only looking for 1, 2, 3 because they are looking for the nucleotide. Those weak hydrogen bonds, you can talk about them when you're talking about the whole structure of DNA and the whole structure of RNA. The next difference is the base. RNA nucleotide has uracil. So adenine, guanine, cytosine, uracil. While DNA nucleotide has thymine. Thymine, adenine, guanine, cytosine. Whatever order you write is correct. You don't need to cram as long as you know those 4. Is it clear? You know when you have too many batteries, né? They start to go down, 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 down. And then after that, you got other people to come in. All right. There. Hola. Because that. Now you did close. Sorry, what did you eat? What did you eat? All right, let's continue. All right, we're done with DNA, eh? DNA replication. Here, I'm not going to take yes. Yes, we are going to DNA replication. What is DNA replication? I want to see the word exact copy or identical copy. Doesn't stop there of itself. Is it clear? If I don't see that, 0. Is it clear? Okay. When does it occur? When does it occur? During interphase or before the cell divides. Remember, interphase is not a phase of meiosis. How does it occur? Let's leave it. Why does it occur? Why? Why does it occur?
To double the number of genetic material.
Skanta, coming to you. To double the genetic material. To double the genetic material. To double the chromosome number.
So it also says
Yeah, to double the chromosome number. Most. When guys, I told you, do not eat immediately. Why? When you eat immediately, the blood is gonna be drained from the head to the stomach, and then what? Oh, you just tasted. You just tasted. So that's your snack, right?
Yeah.
All right. We said, why does it occur? To double the chromosome Number. Nikki, make sure that you don't mess me up. Ne? Make sure you don't mess me up. Ne? Yes. Lastly, where does it occur? Faith. In the nucleus. In the nucleus. Where do we find the nucleus? In the cell, ne? Do all cells have nucleus? Okay. Okay, all eukaryotic cells, they have the nucleus. Lastly, how does it occur? I need somebody here to come and just describe for me. When I tell you to come here, I want you to tell me how does DNA replication occur. Those are some of the processes you might not miss. Because even if I say it, it's going to be exactly the same in the book. You understand? Now I want to hear from a student because this is a process. And the process cannot be changed. It's like eating. You can't say that today you are eating from this side, going this side, and then the other day no, it's a process. Mouse? You understand? Yes. So who can describe for me how does it occur? The process of DNA replication. Come and then just tell us.
Can't you just say it from
What's about from here?
You know when you don't select yourself, I'm gonna pick you. You know that.
Can I bring my book now? Okay. just for you.
Stand here.
Yeah.
Yeah. So guys, DNA replication now. Okay. So first, the double helix unwinds.
Nice.
So must I write it or can I just explain?
Explain.
Okay. The double helix unwinds, and then the weak hydrogen bonds break to form two separate strands, each acting as a template, and then free-floating nucleotides are used to create a duplicate of each strand, whereby adenine pairs with thymine and guanine pairs with cytosine. And then when two new strands are formed, So now there's 4 strands, and then there's one original and one new strand for each. Is that okay?
We are listening.
She's done.
Did you see that she's done?
Oh, this process is controlled by enzymes. Yeah, that's the last one.
Okay. This process is what?
Controlled by enzymes.
This process is controlled by enzymes. All right, thank you, Madam who? Madam Laika. All right.
I have nothing to say.
Can I just say that?
Okay, someone is asking, I can't see the body. All right. Another is saying that I can't see what is not say. Okay, which topic? Hey. All right.
That's the gold one.
I knew that. I knew that.
Okay, guys. Ne? Simon. Okay, someone is asking, you can't see the body nicely, but you don't worry, the person who is there. Thank you, Laika, ne?
You're welcome.
You missed something.
Okay.
You missed something.
Genetically identical.
They are genetically.
Identical. Yes, I knew it.
That's
Yeah,
number
okay.
one. Where do we find the nucleotide we use?
In the
Yeah,
nucleotide.
Which kind of nucleotide?
Each one.
DNA nucleotide. Eh? Yes.
Oh yeah.
Now,
You are.
I want you to write something so that you know they will be saying what to write.
Don't worry, I will speak when I'm writing.
You are cut. So we can't see you.
You can't see me, ne?
You are cut.
Yeah, it's
Like it's zoomed. Yeah, it's zoomed in. It looks like it's.
Alright, it's fine.
Someone was complaining about not seeing the words, isn't it?
Yeah, it's the ones.
That's.
Alright, it's fine. We are saying that the next thing is what? How? Please, when you're describing this, you have to say that using free floating DNA nucleotide. We want to see free floating DNA nucleotide. The word nucleotide of DNA, it must be there. Why? Because when we go to transcription, we shall say that using free floating RNA nucleotide. Is it clear? Sisonke? Yeah?
Who?
Sisonke?
This one or me?
Sarafati, you
The
can
one
answer this
behind
one. There. All right, let's continue. Can you wrap it up? The process of DNA replication? Can you wrap it up together?
Yes.
Eh?
We'll talk about how we can
Yeah, let us look.
double helix unwind,
Double helix unwind,
weak hydrogen bond break,
weak hydrogen bonds break
each to form two
to
stems,
form two strands.
each acting as a template,
The word each is very important. Each acting as
a
a
template,
template.
using free DNA nucleotide from the nucleoplasm.
Using free DNA nucleotide from the nucleoplasm,
A complementary DNA is formed
a complementary, the word complementary must be there. Complementary strand is formed. Where?
when a adenine is
Adenine, where adenine pairs with
thymine
thymine and guanine pairs with cytosine.
and two new DNA.
These are called what?
formed.
Complementary base pairs. Ne?
Yes.
Two
Two
new DNA are formed, which are genetically identical. One with original strand
and the other one is new.
and one new strand. Is it clear? Yeah, this process, it has enzymes. But, we don't talk about the enzymes now. Is it clear? So we are done with the DNA. So basically, can I have my bag here so that I can get more of? All right, sister. You know, yeah. People are eating a drink. How do you call it?
No, sir, I need one.
Red Bull, Monster,
I want to
Dragon. Why energy drinks have those names? Dragon, Monster, Red Bull, everything. Hush. All right. Let's go to another question. This one, it can come in objective or any other way. If adenine is 20 What is the percentage? I know this one I've taken time without calculating it. What is the it's easy, but you forget. If DNA is 20,
DNA.
sorry, if adenine is 20%, what is the ratio thymine to guanine?
2 to 4.
What is the ratio of thymine to guanine?
2 to 3.
Yeah, it
2 to
is.
3.
Yeah.
You think this is dating? You're just guessing, eh?
No,
It is 2 to 3
if A is 20, then
20%
C
is
is
to
20,
30%.
then it's 60.
You like to guess, eh? All right. Who can come here? Who can come here and do it for us?
Like again.
Kamgero?
I didn't say I wanted
Kamgero?
to.
Woza.
Kamgero?
Okay. A is 20.
Yeah.
Lord, I have many questions. And then, if this is 20%,
That means
remember our Bible. When I say Bible, I mean in this period, what we use. Ne? A goes with the T, isn't it?
Yeah.
G goes with
C.
C. These are complementary, isn't it? If this is 20, definitely this is also 20.
Yeah.
But they say this is percentage, so it's out of 100. So 20 plus 20 gives me 40. But the total is 100 because it's percentage. Is it clear? So it's gonna be 100 minus 40 gives me 60. Is it clear? But 60 is for 2. So I divide by 2 and I get 30.
So we
Is
were
it
guessing
clear?
this one.
So now what I want is thymine to guanine. Thymine is 20, guanine is 30. I have to reduce it to the smallest value because the ratio is supposed to be expressed the lowest value so that you don't get a fraction. So times divide by 10, divide by 10. Is it clear? What's the answer? It's 2, 2, 3. You know what they can bring in exam? You know what they can bring in exam? Huh?
Yeah.
A, 3 to 2.
Yeah.
B, 2 to 4. C, 4 to 2.
So you're gonna ask for us.
D,
Calculate.
2 to 3.
Oh, you must work it out.
Do you understand? They bring it objective, but they bring, you know that they're gonna be 2 and they're gonna be 3. You have calculated it because you have seen 2 and 3. And they said 3 to 2. Because he's on A, you don't want to read what is on B and what is C or what is on B. You suck on A, you get it 0. Because they say the
Diamond to guanine.
thymine to guanine, not guanine to thymine. Are you seeing? Yeah.
So who was guessing, sir?
No. Guessing. Why am I saying it's guessing? Is because I need to see the working.
But we were done.
What's that
It
side?
was done.
Huh?
The working was done.
Sorry, play on. On my door closed.
10. That's good. 10.
All right. Can we continue? Can we continue? I think we're done with DNA. Protein synthesis.
So profiling
DNA profiling, guys. Do you do that one? You know.
for revision.
That one you know. Let's just do it by tips. Ne? DNA profiling by tips. DNA profiling, the definition, they scooped it out of the exam. But it's a technique which is used to identify someone's DNA profile using a non-DNA profile.
Yeah.
Someone who you don't know. But you have another one you know. So you compare. Technique used to identify someone's unknown DNA profile using a known DNA profile. I know the child, but I don't know the father. You understand? Therefore, what do? I bring the DNA of the child because I know the DNA profile of the child. And then I compare it with these 3 fathers. And then I find out who is the real father. Is it clear? Yes. Sister, don't cover your head. Don't cover. Hey, Wena. I saw you when you were eating. Sister.
Yeah.
Let's continue. Give me some of the doubts of DNA.
The what?
Some of the what?
The debates.
Debates.
Yes. Or the doubts.
Wait.
The
Very
disadvantage.
expensive.
Oh.
Expensive. How much the
Oh.
Yeah. Do you remember that?
Yeah, Michelle.
Uccelli va per la sera.
I'm gonna take you.
Voglio scegliere di piacere. C'era come da anche molto Dica,
Okay. All right. Hey. Sorry. Hola. Do you respect me? You want me to give you a special room where you can sit and you relax with your friend? Is that what you want? I'll keep you there until morning. Joke with me. In fact, we are here for business. What is the business? It's your life. You understand? If you lose it, the business is gone. All right, let's continue. Hey, why is DNA profiling not important?
Not important.
Yes, that's what I'm saying, that some of the disadvantages, some of the debates, we can bring in a different phases and then
Human errors.
human errors. Ne?
Yes.
Okay.
It's expensive.
It's expensive. Ne? You may not be unique.
So false evidence.
False evidence. Framing.
Yes.
Yeah, they can frame you. Ne? Yeah, a lot. You understand? All right. So let's continue. Protein synthesis. How many phases in protein synthesis? How many? Two. The first one, transcription. The second one, translation. I need someone to describe transcription here. I need another one to describe translation here. And I'm going to pick you from the back side. Ne? Yes. The back side. Yes.
Say it nice.
I should say it nicely. So I should say how? How should I say it?
How you say it to the class?
At the back. The back, yes. All right. Who is ready?
The back.
The back lady.
Faith, come and tell us what you have in your head. Huh? You're not ready. Nikita, you better sit down. Ne? You better sit down. I'm giving you 1 minute. I'm giving you 1 minute. And then after that, I choose someone to come and describe for me. Who is ready? Why what? No light, you want to be the darkness? Oh, are you the eternal animal? That's you only see at the darkness. People are nocturnal. Hey, all right, who is ready? Who is ready? Kelly. Hey, who is ready? Yes, come. No, come here. Come here, Michelle. Come here, Michelle. No, whatever you give us is right. We correct it. Don't worry. Michelle, 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 Michelle. Michelle, come here, come here, come here. Get the marker, get the marker. Get the marker. Michelle, come. Michelle. Guys, I'm not continuing until I see a student on the board. Yeah, you can come. Come. Transcription. If I don't receive a student here, it means that I've not been teaching you. Yeah, that's the meaning. That's the meaning. That I've not been teaching you. Transcription. And then be ready for translation. Be ready for translation. All right. Guys, don't mess up my name. Okay, let's start. Transcription, sir.
So we know in transcription, that's where the DNA will turn into the mRNA coding. So we know
Can you use that small speaker so that yes.
So we know that in transcription, the DNA double helix will unwind.
I'm gonna ask you. I'm gonna pick someone to go back and describe what he has described.
Yeah.
You understand?
It's right.
Start again. Start again.
So in transcription, we know that we want the DNA coding to form an mRNA, a messenger RNA coding. So we know the first process that's going to happen is that the DNA will, the double helix of the DNA will unwind. And then we know that even when the double helix unwind, the weak hydrogen bond will break and form one single strand. So if you know that when the DNA double helix unwind, they will form two separate strands, you know, and then the single strand will form as an mRNA. It will form as in a template, you know, for mRNA. So when it forms as a template, the free nucleotides from the free floating mRNA nucleotide from the nucleoplasm coded for mRNA, where the complementary mRNA changes to A to U and G to C, something like that. The process will happen, something like that. So this whole process, when all this whole process happens, we namely call it a protein synthesis. So what will happen is that the mRNA is going to form from that's why I really that's why I know. So.
Now some people are laughing at him, yet
And she didn't want to come and
Imagine
explain
Michelle.
what was that.
Michelle.
eh? Yeah, someone else wants to help.
Wait,
But thank you very much. At least you have tried. Yes.
and then guys, after that process, you said it's called protein synthesis. First, the mRNA gets the code from the DNA to code for protein synthesis to create the codon, and then the mRNA strand moves from the nucleus through the through the nucleopore. No, from the nucleus through the nucleopore into the cytoplasm and attaches to the ribosome for preparation of translation. Oh wait.
Guys, let me ask. If this question comes in exam, how many of you are going to pass it? Huh?
We're all learning, sir.
Yeah.
If this question,
After tonight, so we must all pass it.
eh?
Yeah. After tonight, we're all back.
So can I
After
try
the
translation? So can I try translation?
Okay. Guys, don't forget DNA double helix, unwind. Weak hydrogen bonds, break. Weak hydrogen bonds, breaking is the same thing saying unzipping. To form two strands. One acting as a template for formation of a complementary strand using free floating RNA nucleotide from the nucleoplasm. Is it clear?
Yes.
And then, whereby adenine goes with uracil, and then cytosine goes with guanine. Don't forget those complementary base pairs. And then a messenger RNA is formed, which moves out of the nucleus via
The nucleopore.
to
The cytoplasm.
on
Attaches to the ribosome on the ribosome.
the ribosome. Simple. Come, Carol.
Not that simple.
Who is next for
Can
translation?
I try? I'm just trying.
You can
I
come.
don't want to say.
Sister. Sister.
I'm trying. I don't know it, but I'm trying.
Okay, tell us.
Each
So you must write what she said. Throw it.
each DNA carries a specific amino acid.
Each RNA?
T.
Oh, each tRNA.
And then when the anticodon.
Guys, you have never heard about that. You have been saying,
When the anticodon of the tRNA matches with the codon of the mRNA, a specific tRNA amino. No, the tRNA brings a specific amino acid to the ribosome, the required amino acid to the ribosome, and then I don't know that last part.
What you form when you bring the required amino
A
acid to the
peptide
ribosome?
bond. Peptide bond.
No, a bond is joining something. So what does it join? No, it must be.
But I tried.
Yeah, you tried. Thank you. But I'm just reminding you with the different ways so that you
Required protein.
guys, you're writing when?
Tuesday.
Count it as Monday. Count it as Monday.
Wait, so they're guessing what our timetable is.
Count it as Monday because Tuesday don't have time.
Oh yeah, okay.
Yes, count it as Monday.
Can't say mine.
Guys, how do we mark this? You can phrase it the way you want.
Okay.
As long as you talk about the following marking points. Is it clear? According to what?
To what exactly?
The Bible.
Excelling life science.
According to huh? According to the movies.
Yeah, definitely.
According to guys, you have when you are phrasing it, you can phrase it with the messenger or with the transfer. You understand? According to transfer RNA, anticodons, or you can say according to the messenger RNA codons. You understand? So you saying that according to the messenger RNA codons, you saying that messenger with the codons is a tick. So you want to see the word codon versus messenger. According to the messenger RNA codons, number one. Are you saying? They transfer RNA not just with the complementary. Want to see the word complementary or matching anticodons. According to the messenger RNA codons, yes. The transfer RNA, what does it have? Complementary. What is complementary there? Anticodons. According to the messenger RNA codons, the transfer RNA with the complementary anticodons bring the required amino acid to the ribosome. Is it clear? Then, what happens when the amino acids are brought? The amino acids are joined together by peptide bond to form a required protein. Simple. Can I say it again in less than a minute? According to the messenger RNA codons, the transfer RNA with the complementary anticodons bring the required amino acid to the ribosome. The amino acids are joined together by peptide bonds to form a required protein. Simple.
Okay, so what's
Let's see how do we mark. According to messenger RNA codon, messenger RNA codon tick. The transfer RNA with the complementary tick, ne? Anticodon tick. Bring the required or the matching amino acid to the ribosome. Bring the amino acid to the ribosome. Tick. Amino acids are joined together by peptide bonds. Tick. Guys, don't confuse the peptide bonds between the different amino acids with weak hydrogen bonds.
Okay, so with what I said, how much was I gonna get?
How many marks?
Yeah.
If you
what I
I was
said.
supposed to mark you, you're 50%. Don't say at least. Don't say that. I don't want that at least. I want all of it.
But I tried.
I didn't say that you didn't try.
But I'm just-
You did better.
Yeah, better than Michelle.
Don't compare yourself with someone else. You know why? You know why?
I'm just
Different questions,
I was
different
just-
mangoes and oranges. All right, let's continue.
like mango.
Please put your star on transcription. Don't go to the exam without knowing transcription, without knowing translation, without knowing the structure of DNA, without knowing DNA replication. Is it clear? Lastly, on DNA, it's about the mutations. But mutations, I prefer when I'm showing you the diagram.
Yeah.
But let me just hint about it because I have some questions I'm going to give you. Let me just hint about it. Then, when you will talk about it in the paper there. Don't forget that if a mutation changes, a base is changed. Ne? And when the base results in a different amino acid, then a new protein is formed. Is it clear? For example, for those who were in not my class, ne? Or for those who were in my class, but the body is in my class, but the mind is outside the class. Whereas, yeah, those things happen.
Yeah.
You can sit with someone goes home, goes to the toilet, does whatever he does there, comes back.
Come back.
Hey, dunia. All right. For example, I'm just gonna give you an example here, ne? You have 3 amino acids, ne? Glycine. Well, let me say methionine. Leucine, tryptophan. Ne? And then now I have A. I'm just gonna make them to be simple, ne? And then I have A, G, A, ne? Then I have C, G, C, A, ne? And then I have the third one, AGC. Yeah? Yeah? The arrangement is different, eh? And then AT. Okay, it depends on which kind of amino acid base I've used. AC. Is it clear? Now, how is the mutation? How am I gonna know that this mutation is gonna change the structure of the protein, or this mutation will not change the structure of a protein? Remember, sometimes this is given. The first thing you must know, is read what is on top here. They will say these are amino acids, correct? But read what is here. Ne? They can say DNA or RNA or, you understand? Transfer RNA. Is it clear? So if they say messenger RNA, now they say DNA. You have now C. No, you have, yes, C, G, G. These bases where one base was changed. G was changed to T. Is it clear? That when you read the question, they're saying that a mutation occurred and the base, this base, the third base, was changed to T. So it means that the new code I'm gonna have is gonna be CGT. What will happen to the protein of that individual? What will happen? What do? You read it. Remember, this is DNA. Even if I don't say that this is DNA, because of the T, it is enough to show you that it's what? DNA. But this, what is here, is RNA. If you are RNA, so it means that I'm gonna say G, C, C. Which kind of amino acid is that? I come here. It's gonna be leucine. I see. But I said now, it changed the base to T. It means that now I'm gonna have G, C, A. I see. I come here and I find out what is the base. What is the amino acid? It's gonna be still leucine. So it means that this mutation will not affect the functioning and the structure of that individual. Is it clear? It will not affect. It will remain the what? It will remain the same. That's the meaning here. Is it clear? Some people are saying, "Ooh, yeah." When people, who is feeling cold? I told you that here, there is, the pressure will be heavy. There is cold here.
You're just tired.
You are tired, eh?
Yeah.
So I'm seeing the kind of students I have. What time do they sleep?
So you sleep now
You
and then
sleep
wake up
early
later.
and you wake up at 12.
Generally, that's what
Okay, lastly, look here. What about a mutation is like this? Begala. What about when a mutation is like this? Huh? What about when the mutation is like this? Now, it was like this, ne? And then now this does not change to that. Now I get, let me call this one C, ne? So that is simple. All right. Now it means that this one is gonna change from C to T. Is it clear? So if this one changes to T, then I'm gonna have this. So what will happen to the protein? That means that when I read this, it's gonna be leucine. Ne? But when I read this is gonna be this one is gonna be A, it's gonna be C, it's gonna be C, isn't it?
Yeah.
So it's gonna be tyrosine. The first one was leucine. Now, what does it mean?
There's a change in.
There's a change in the protein. So it means that the change in this base from this to this will result in the change in the structure of protein formed and the functioning of that organism. Will also what? Will also change. Imagine in the morning you wake up with the 2 horns.
So that's now a mutation.
So who has ever dreamed about when you are flying?
Me.
Can you glitch?
Who has ever dreamed? Yeah? You have never dreamed about when you are flying, eh?
You dream about when you're falling, sir.
Yeah? Or when you are falling.
Can you jump?
You
Yeah.
only want to dream about nice things. Alright, this marks the beginning of the end of DNA. Any question? So if it is RNA, it means that you have to change 2 times. Yes. Huh?
Yeah.
You have to change it. If you have, for example, A, if it's TRNA, if TRNA, what happens on TRNA is what happens on DNA.
But it's just.
So it's gonna be A. Eh? So this is DNA. So it's gonna be. Remember, A goes with.
No, you.
Remember, this is messenger now.
Yeah.
It's gonna be you. But you have to change it to TRNA. You goes with A. I say that.
So it's
In the most cases, what you see here is what you're gonna be here. If this DNA and this DNA, you don't change, you read it direct. Yeah. Remember I told you, our guideline is supposed to be DNA, RNA, tRNA. Yeah. Then transferring A, and then amino acid. So it must be like this. Sometimes they can give you this, they don't give you this. They give you this, they don't give you this.
Continuous control.
Yes.
Right done.
How many minutes do you have?
You still have like almost an hour, so like 45 minutes.
Wait,
You still have 45
wait,
minutes.
it's break.
How many minutes do you have?
Past 9.
Meiosis.
You have.
Can we start meiosis? Meiosis is gonna be a little bit faster, ne?
So wait, how many minutes is our break?
15 minutes.
Don't worry. We'll talk about that, ne? We'll talk about it, ne? Guys, is it clear? Can I give you a test now? Okay, remind me. Brainstorming questions. Remind me. Bonds between amino acids. Guys, I don't want you to delay. Peptide bonds. Yes. Peptide bonds. When I say bonds between amino acids, they must be here. When you delay,
You're also saying bones.
okay, can I repeat? One. Okay, we are going. Bonds between nitrogenous bases.
We kind of just want to say that again. Write it like, are you saying it so fast?
Some people they are 18, but they have gone back to 2 years.
I saw wait.
All right. Okay. Weak hydrogen bond. Is it clear? What's the delimitation?
A change in the base of
The base of the bottom. The base of the bottom.
when the base of the DNA or mRNA or RNA changes, yeah. When the base changes, yeah.
Sudden change.
Something like that.
The base of
Yeah.
DNA. Or in a nitrogenous base.
DNA or
Bravo, I'm coming to you. Question. Usisiwe. Don't forget that you did cheat me. Usisiwe. You're supposed to come here and present. Usisiwe. Don't frown. Don't frown. You know some people when they frown they look beautiful. Please don't frown. I don't want to see some people here. Alright. Question.
Yes, sir.
Process which occurs in the nucleus. I want you to know the processes.
DNA replication.
DNA replication. Only
Your transcription.
and transcription. DNA replication and where does DNA found?
In the where does DNA
Where,
found?
yes,
Where does it found?
yes.
Okay, in the nucleus.
3 locations.
Nucleus.
People are changing their faces.
Yeah. And then
In the mitochondria,
cytoplasm.
chloroplast. People are just remembering. Yes. All right. Lastly,
Yeah.
give me 2 locations where we find DNA in plants. Give me 2 locations where we find DNA in plants.
In the chloroplast
Mitochondria.
sometimes.
Huh? When I ask you in plants, all the 3 locations are found. When they ask you in animals, only 2 locations.
Which is
Nucleus.
And mitochondria. You have forgot
But in the plants, talk about the nucleus, talk about the plants, talk about
mitochondria.
So you can't because it's late now. No.
lastly. Yeah?
No, but I
Okay. Okay, you remember. All right. What is meiosis?
So they refer to
Nikki, stand up. Busisiwe, go where Nikki is. Nikki, go where Busisiwe is. Hurry up. When you mess me up, we start the story. That's so nice, no? Guys, we are here to study, no? Not to mess up each other. Sister, don't complain, don't do anything, just shift. All right. Hey, you are saying anther produce pollen grains. Pollen grains, if we bring these are plants. If we compare it with the human, anthers are the testes. Pollen grains are the sperms. Clear? And then ovary is the same thing. Even in plants are called the ovary. But in plants, they don't produce ova. They produce ovum. While in humans, they produce ova. It's a lot. It's one ova.
Thank you.
That's it. I'm still waiting to these people to swap. Nikki, if you don't want that, you're gonna come here. Nikki. Nikki. All right. Huh? What is meiosis? Meiosis. Guys, meiosis is gonna be fast. What's meiosis? The reason why I've spent more on DNA because we started DNA in term 1. You understand? And then as we are continuing, you're gonna be a little bit faster because this content we have
Be
is
slow by the nervous system.
nervous system. We're gonna be faster. It's simple, easy. You're gonna see it. You're gonna eat it like
Menstrual cycle can be please.
All right. What is meiosis?
It's a type of cell
The
division
type of cell division that results in 4 daughter cells with each having half the chromosome number of the parents. Yeah.
Type of cell division which results in 4 daughter cells, each having half chromosome number to the parent. It means that these cells are not genetically identical. Or otherwise,
They would have.
we would have looked like clones. Remember that question which is always being asked? Are you sure your father is your father?
It's only you that's asking that, sir. Nobody else. Don't worry.
Hey, meiosis. Meiosis can make things happen. You resemble the neighbor. Hey, and your father doesn't like the neighbor.
Your father likes the neighbor. Come here, man.
Come here. Come here. Are you okay?
Yeah.
All right. Give me this. You need to know, eh? Benefits of meiosis. Why meiosis should occur?
One.
Number 1,
Genetic variation.
genetic variation.
Higher chance.
Guys, there is the first answer I need to hear.
Which is
If meiosis is not there, we can't exist.
Oh, yeah.
that's why I said higher chance.
There is the first point I need to hear. The function of meiosis to produce the gametes.
Yeah.
To produce. Before you talk about variation, you talk about what. Even if variation is not there, we can exist. But if we don't have gametes,
We won't exist.
if your father, my father, whose father whatsoever did not manage to produce sperms, and your mother, my mother, whoever did not manage to produce the ovum, we wouldn't have existed. You understand? So please, stay what?
Virgin.
Please use our things sparingly.
So does the do they find
I'm done.
also say sex before marriage?
Huh?
Sex before marriage.
The Quran doesn't. It doesn't.
No, it's not
It's the what?
You guys have, you can.
It's the what?
Stay virgin.
It's the what?
So it means that it allows you to have
This one said he's tired, but when talked about sex, he's awake. Alright, produce gametes. Ne? That's number one. Number two, genetic variation. Number three, having the chromosome number. Remember, we started with 46? Ne? No, man. We are diploids. We are diploids. 46. When you say 23, there is something which is missing. The word pair. When you say 23, you must add this word pair. If you say 46, sister, I'm teaching life science. I've not talked about pizza. All right. So we have to have so that remember you form 4. You form 4. Each with the haploid. Is it clear? So 2 sets of chromosome, 2N, 2 sets of chromosome is called what? Which term do we give it?
Haploid.
Diploid. And then half
Haploid.
haploid. Is it clear? Half means half. Di means
Double.
so when it's double, it is 46. So we are diploids. But our sperms, your sperms, their sperms, they're haploid. You understand? Yes, they're haploid. And the same thing over. And what? So that the haploid of the sperm, 23, and the haploid of the ovum, 23, they fuse again in a process called fertilization to form a diploid again. 23 pairs or 46 chromosomes. Is it clear? Guys, I'm saying these things, but people will be remembering. You know what they will be doing? Looking at the body up, the walls, as if the answer's there.
Yo,
There's a better way.
it's calling me. Yo,
All right, let's continue. Yeah, we're gonna have that interference just a bit until around 10:30.
We're going to dance.
No man, I'm saying
Yeah.
interference. All right, can we continue?
It will wake us up though, for real.
Yeah.
They can ask you a question. They asked this question, I think 2019. Describe the karyotype of a male human. A karyotype of a male human. Describe the karyotype of a male human. Who can describe for me? Guys, everything is here, guys. Don't do this to me. And I told you everything very well. A karyotype of a male human. Bone. Yeah, once you see this, it's a karyotype. How do I differentiate that this is a male, this is a female?
It has XY.
Please look. No, they won't say this XX, this XY. I'm saying on the picture. Look at chromosome number
X is long.
23.
Okay. If it's 2 of the same length, then it's a woman.
Yeah.
And if the one is short, then it's a guy.
You understand?
Yeah.
When the 2 of them of the same size,
Like if
then it's a female. It means that the same.
this one is the size.
And then if one is longer than the other one, then it means that that is a male. Remember, X chromosome is bigger, longer.
So then do we just
But-
write that?
Yes, but no, I'm just trying to show you. And then the Y chromosome is shorter. So how do you describe a karyotype? A human being is made up of 46 chromosomes or 23 pairs. From chromosome number 1, chromosome number 22, pair, those are pairs. From chromosome pair number 1 to to number 22.
We call them.
Yes, we call them autosomes. Autosomes. The 23rd pair is called 23rd pair is called gonozome.
Yes. Or
Eh?
6 chromosomes.
Or 6 chromosomes. Eh? This pair can be is made up of X and Y. Is it clear? Whereby if X and X is female, and if it's X and Y
It's male.
is male. Is it clear?
Yes.
Yes. So these chromosomes,
X is longer.
X is longer than Y. So basically, you just look at that diagram and then you describe. You know, this thing of description, you must have a picture in your head.
So if we draw on the paper,
It's fine. Don't even rub off. Just
Just a
give
small
them their
thingy
paper.
there in the corner.
Sister,
Yeah.
don't waste even your time rubbing off. Just do your sketch work. It's fine. Make it dirty. Yes.
You've seen my
When I say paper dirty, say yeah. That's why I always tell you that if I see your book clean, I will say check this person. He's just playing the book. How can the book be clean? How can a book be clean?
So, but then we must pay for this thing. I must pay for this thing.
How can a book be clean if you are reading it?
It's dead.
Come here, explain that to me. Come here, explain that to me. How can a book be clean for a student?
You take care of it.
When you are reading it, you know when you take care of it, you put it in a what? In a safe.
Shell. So, but you have to always look at this. I'm paying for you. Look at this. See, even the other back is black.
Sister, whether I gave you the book or I didn't give you a book, I want to see that the book
It's broken.
is being used.
Mama said, even you want it back.
Yes.
Exactly. And if
Yeah.
it's broken, you have to pay.
It's broken.
I didn't say that. I said if it's lost. I said yes.
Okay.
But don't be
Careless.
don't be a person who doesn't have responsibility.
Don't be a cheater.
Make sure that you read it. Yeah. All right. Stages of meiosis.
You all.
Stages of meiosis.
No, interface.
Interface.
Prophase 1.
I'm about to change you, ne?
Metaphase.
Interface.
Prophase. Metaphase.
Interface. Speak. What is it?
I said prophase.
Huh?
I said prophase.
No. There's something there. What is it? You're tired. You are tired. In fact, you are a soccer player. You play for how many hours in the game when you are running? One, eh? Is it one? You know how much energy you are using? And you come out even here. Yeah. Make sure that make a mistake and you win. How much energy will be having? What if your statement comes out with 7? You think you're gonna remember this? Guys, there is no one who is successful who doesn't put in. Forget about it. If you don't want to put in,
Put in, get rid.
no, it depends on which kind of success you want. You understand? Even, let me tell you, don't do this. Even, let me leave it. Even the so-called, I don't know them, but I've ever heard about them. Even the sex workers, they make their face beautiful.
Yes.
So that they attract, they also put in. You understand? But when I just want to sleep there and then Max come to you, guys, things don't work like that. Okay, let's continue.
Let's go.
Remind me, if
The phone.
If not,
match, which means
Interface.
interphase. This is not a phase of meiosis, no?
Prophase.
Prophase,
Metaphase. Anaphase.
phase, no?
Yeah.
Okay, I'm not going to ask you what is happening here.
Okay.
No, because you have to read that. Yes. But I have a question here for you. When does crossing over takes place? Which phase? Prophase 1. When you say prophase, 0.
Prophase 1, yeah.
When do chromatids move to opposite pole? When do chromatids move to opposite pole? Anaphase 0. Anaphase 2.
Yeah.
Ne? When do cytokinase occur? Telophase 1 and 2. What is cytokinesis?
When the cell divides, the nucleus.
Karabo, I don't think you are in my class. Karabo. No, Viva.
Is it you? Oh.
Rendan, are you in my class? Serious? Across two chromatids crossing over. How do you call that point? Where two chromatids cross over. I'm asking Rendani. Chiasma. What is the plural for chiasma? Plural for chiasma. If there are so many chiasma, how do you call it? What did I say? Chiasmara. Chias what?
Chiasmara.
CHIASM
A-T-A or R-A?
Huh?
S-M-A-T-A or R-A?
A at N.
So if I spell it wrong, it's exam.
Huh?
Mara or Mara?
What's your name? What's your name? Lonatemba. Luanatemba. Hey, are you hearing what? You don't want to be spelled wrongly, but you want to spell.
No, so I don't want to, but when it's like.
Forget about that and memorize what is correct, so that that weakness is out. If it happened, it has happened, but make sure that it's out. You know, sometimes if you have that loophole, you end up. Lastly, when does a bivalent form? When does a
In the middle
bivalent?
of crossing over. Yeah.
Huh, people.
In the middle of.
When does a bivalent form?
When centrioles move to opposite poles.
Oh, no. Which phase?
In the middle of crossing over chromosomes and virium.
Okay, what is a bivalent?
I don't know. I just know this is what notes are. Yeah.
What is a bivalent? Litabo. What's a bivalent? Isn't I have another litabo? When does a bivalent form?
In the middle of but I don't know what the bivariate is.
Huh?
Some, yeah. During crossing over.
During crossing?
Yeah.
Oh, man. So what's a bivalent? Homologous chromosomes coming together. When the homologous chromosomes are together, they form what you call a bivalent. Bivalent.
Oh, like it's in bind.
Yes. What is non-disjunction? What is non-disjunction?
Failure of.
The mutation that causes Down syndrome.
Yes.
What is non-disjunction? Blairwa, what's non-disjunction?
The failure of chromosomes to separate.
Zwane, what is non-disjunction?
The chromosomes are equally dense on the nucleus.
Guys, I'm going to ask you to shift to this side because you are too far. I don't think even you see. So the next class, when you break, I want you here so that I can see you in the eyes properly.
See with eyes.
Cabello, what is a what did I say? What is now this junction? Huh? Chromosome?
So can you say like the failure of chromosomes to separate?
Failure of the chromosome to separate when? During anaphase. Failure of chromosomes to separate during anaphase. It can happen in anaphase 1, it can happen in anaphase 2. When you say in anaphase 1, then you can say in anaphase 2 or in anaphase. Is it clear?
Yes, sir.
Remember in anaphase 2, it is chromatids. You know, chromatids are supposed to separate. You understand? But we can also call them sister chromosomes. Remember when they reach the core, we call them chromosomes. You don't call them chromatid anymore. If you check that book, I indicated it nicely. Sister chromosomes. We call them unreplicated chromosomes. Chromosomes with the single-stranded chromosome. So during DNA replication, it becomes double. Hey, this one we can call it double-stranded chromosome or replicated chromosome. The one which is single, as long as it has this, is a chromosome, not a chromatic.
Chromatid when it just separates before it reaches the core.
Yes.
So if I say the chromosome when it has reached the core, then I'm wrong.
You are wrong. We only call it chromatid when it's moving in anaphase. When it reaches the pole, it's chromosome. When you say chromatid there, we cross it. Is it clear?
Yes, sir.
What's the difference between cytokinesis and the karyokinesis?
So karyokinesis is the division of a cell, and cytokinesis is the division of the cytoplasm.
Someone is raising the hand there. Thank you. And the karyokinesis. Guys, remember I said karyotype. Which kind of nucleus do you have? Karyo. Whenever I talk about describe the karyotype of the human, it means that you're describing what happened in the nucleus. You remember, I said that when you talk about karyo, you're talking about the nucleus. Please don't forget about that. So karyokinesis is division of the nucleus. Cytokinesis, division of the cytoplasm. Is it clear? Yes. Give me two differences between meiosis 1 and meiosis 2. Two differences between meiosis 1 and meiosis 2. Hurry up, guys. Has homologous chromosome? Which technique did I tell you to use to remember? Go to each phase. In prophase, you'll find out in prophase 1 there is crossing over. In prophase 2, no crossing over. Go to metaphase. Homologous chromosome align along the equator in pairs and randomly. The other side align along the equator. Chromosomes align along equator singly. Guys, must remember that. Yes, anaphase, chromosomes move to opposite poles. There, chromatids move to opposite poles. Telophase, 2 cells are formed, 4 cells are formed. So, guys, don't forget about that. Look clear. Don't forget about it. Meiosis 1 is it reduces the number of chromosomes. While 2, it maintains the number of chromosomes. Isn't it? Because meiosis 1, you start from Shoni, You see me? Me? Listen. Oh. So if you were short like me, You just do like this, you don't see. That is what you are told. Eh, by the time you reach here, we see you. We see you, baby. Okay, guys, is the water there, né? So that she prepare the coffee. All right, I want to finish this, né? So that we do some few questions about it, and then we move to the next one. Reproduction in vertebrates, we're just going to talk about the way we are talking here. Reproduction in human, we'll talk about it like this, but the processes, we'll discuss them here. You understand? Genetics, we have to engage you in. And then, human response to environment.
We're going to talk about it.
This one is the last topic that Nikita, are you checking your face in my class? Nikita? Nikita, you know you are beautiful to change again. Check. No, man. You don't know yourself that you are beautiful. Nikita, you don't know that, ne? You want to keep on checking, say, am I? Oh, there's someone who told you that, hey. Ladies, mirrors. Ladies with mirrors. You see in the men's toilets, you don't find mirrors there. In ladies, because they know you. You the mirror. Here, if we check you stand on the street in the banks of the ladies.
It's better.
Hey. A lady who doesn't have a mirror. Anyway, it's like that. Yeah, it's like that. Ladies, they care about their face. Men, they care about their body. Doesn't matter whether you're ugly or what, as long as it's not a I'm telling you, if you are ugly, I don't know, because it can be ugly to me and you are beautiful to someone else. Or it can be beautiful to me and you're ugly to someone else. You understand? But now let's take it like this. Even if you are ugly, and he's a guy, and you have money.
Beautiful girls.
And don't be ugly in easy. You must be. Yeah, then, don't worry, you'll be happy. The convoy behind you. Nikita, please. The face will be beautified more when the statement has 7. You understand? There is no guy who likes to think. A lady who doesn't think. Think. Thank you very much. They are there. You will go with that guy 2 days. They will kick you like this. Don't worry. So guys, please, ladies, make sure that you sustain yourself. Ne? I always tell you this. In this country of ours, you know, guys, what is going to happen? 20% of the men, they take care of their wives and their kids. The rest, the 80%,
Female black household.
the female will take the responsibility. You don't know, man, whether you're gonna be in 20% or in 80%. So what should we do? Please, let's fix our life straight. Is it clear? Yes. All right. Lastly. Tokeru, do you know what is called Down syndrome? What causes Down syndrome? Non-disjunction. Oh. What happens to Down syndrome? A person who is having Down syndrome. Tell us something about it.
They become slower.
No, I want to talk about genetically. What's happening there in terms of chromosomes?
The chromosome numbers
Huh?
it's not equal.
It's not equal.
Maybe they have like 23 and then the other one is 20.
Listen,
23,
maybe.
25.
Maybe.
No.
Uh-huh.
Infinite.
Okay. They have what?
Unequal number of chromosomes.
Come show me here. Come show me here. Tokero! Alright, Down syndrome.
No.
Uh-huh. Thank you. Guys, people confuse. The reason why I asked you to come and show me something is because people confuse
Yeah.
non-disjunction and Down syndrome. It's not the same.
Non-disjunction may
Yes.
lead.
Non-disjunction causes Down syndrome, whereby non-disjunction, failure of the chromosome to separate, it will result in one gamete having short of the chromosome 22 instead of 23, ne? Or 24 instead of, so this one is short of 1, this one has an extra of 1.
24 instead of 20.
You understand? So if this gamete fuses with a normal gamete now,
That's 47.
you understand? Now the normal gamete is 23. Okay.
Yeah.
So you have 23, 24.
Yeah.
So it's gonna be 23 plus 24, which is 47. Instead of what?
46.
46.
Yeah.
Is it clear? So yeah, that's the meaning that you have an extra chromosome. Listen carefully. Now, the person having Down syndrome has an extra chromosome. Don't just stop there. An extra chromosome on chromosome number 21. It must be chromosome number 21. If an extra chromosome occurs on chromosome number 18, that's not Down syndrome. It's something else. Yes.
So is it possible for someone to have like a slight behavior of a person with Down syndrome?
It's like what?
Behavior.
No, doesn't mean that if you have one characteristic of Down syndrome, if you are weak in maths, and then you say, I have Down syndrome. No, man. You can be having some characteristics, but it doesn't mean that you. It must be a collection of characteristics. Yes. Uh-huh. Yeah, he's asking, does non-disjunction cause other syndromes? Yes, it causes other syndromes, but our scope only we talk about Down syndrome. We have other syndromes, Klinefelter syndrome, Turner syndrome. So
what is that?
but we don't talk about that.
But Down
Only talk
syndrome
about
is on
Down syndrome.
number chromosome 21.
It occurs on chromosome number 21. So how do we bring this in exam? We give you a karyotype. Ne? Ola. O. Nikita, you are about to scam to stand here. Nikita. So whether your mouse, if it is not quiet, what's happening? When your mouse is not quiet, you can't be there. Check now. You made me to forget. Yes. Huh?
Not in our school.
I just wanted to.
Hey, guys, we are focusing on what's going to happen on Tuesday. Hey. All right. Shh.
I don't know what's
It's a fellow.
Come, Hero. What is BB?
What is what?
BB Paris. BB Paris. BB Paris. BB Paris. BB Paris. Come, hello. People are open. What is vivipary?
In the body of the mother.
Amnion what?
In the mother's body is like protection.
Inside the uterus.
The amnion
No.
develops in the mother.
No,
The embryo develops in the what?
in the mother.
In the uterus.
Other nutrients.
It obtains nutrients.
Obtain nutrients from the mother.
And obtains nutrients through placenta. Vivi inside. Nutrients from the mother. Oviparous?
Inside the mouth.
Oviparous is
Oviparous.
Yes.
The egg laid.
Oviparous.
Chickens.
The eggs are laid and the embryo gains nutrients from the yolk. Yeah. We are done.
Sean. Sean. I'm gonna ask you a question. No. Sean, I'm gonna ask you a question. Have you ever tested sperms of any organism?
Why would you do that?
No, I want to see. No, there is a reason why I'm asking.
not doing it.
No, ne?
But so,
Can help. I'm going to ask you another thing. Have you? I'm talking about in the mouth. Have you ever tested a sperm of any organism?
yes, I have.
Sister, have you ever tested? Have you ever tested? Yes. Okay, Natando. Okay, bye. Have you ever tested the ovum of any organism? Guys, how many of you like eggs?
It's an ovum yes.
Making a baby.
Have you ever tested? Guys, some of you don't know the egg of the chicken.
It's a zygote.
It's already a zygote.
Yeah, so it's
The sperm and
sperm and ova.
I'm gonna get
No, you know when you say that.
and when you're eating it. Guys, okay, there are some ovum. In most cases, when the ovum is released,
So they say that people outside want to come in.
people outside it,
Yes.
what kind?
People are late. People are late.
But
We'll tell them at break.
I need to know where they are coming from. Guys, I have a register. Okay.
They'll explain.
Yeah, they'll explain.
Guys, listen. This cross night is strictly life science. I saw some learners who are not doing life science. They are coming. For what?
To chop.
To cheat. Okay. All right. What is the advantage, sister? Go speak from there. What is the advantage of the organism having internal fertilization over external fertilization?
Have more protection.
There's a difference between internal fertilization and internal development.
Yeah, you see, that's what I was asking.
We say that the eggs we eat, mostly, they have the sperm and the ovum. Is the sperm and the ovum, isn't it?
Yes.
It means that they were fertilized before they were released. You understand? So please, don't confuse fertilization and development. Is it clear?
Would nutrition be part of benefits of internal fertilization?
So internal fertilization, the high chances of sperm meeting the ovum.
Wait, repeat that something?
The high chances of the sperm meeting the ovum or fertilization take place. Yes, that is internal. Because the sperms are deposited to a structure whatsoever, either a vagina or cloaca, whatsoever organism it is. Understand? Now the sperms will go direct to the ovum. So the chances are high that fertilization will take place. But external, the ovum are here, the frog is here, for example, is spraying sperms here.
In the water.
How sure are you that these sperms are going to come here? What about that side? What about that side? What about the up? You understand? So there are high chances of fertilization to occur for internal fertilization. Sisonke. Yeah. Don't worry. Don't worry. I'm finalizing this. Lastly, precaution.
Yes.
When they are independent from a few hours after they're born.
Oh, that
How do you know that this is precaution development?
They are young and
If-
able to defend themselves.
they can defend themselves.
After
Are you thinking for the country? Eh? You know when you are thinking for the country, eh? You close the eyes. Eh? You start thinking for the country. Eh? Minier, don't think for the country eh? Now. You will think for the country after. Eh? All right. Shh. Pre. We said pre means what? Prepared. Prepared. Born prepared. Eyes are what? However small they are,
And they got
but they
feathers
are okay.
sometimes.
they are feathers, they are able to.
Have the down feather.
They can walk. Some
They
of them
can
have
walk.
feathers.
They can defend themselves.
So they come out
They
part
can
of the
protect
20%.
themselves.
Huh?
They come out big part of the 20%.
So and then our Trisha, they ask you, give an example of Atrisha. Guys, don't even waste time when you are there.
Chicken.
Yeah.
Babies. Oh,
Why do you think about chicken when you are there?
it's us, bro. It's us.
Humans. They say that give me an example of Atrisha in a bunch.
Animals, no.
No. If they say animals, even
It's
your
still
animal.
humans, yeah.
I'm
Why
a
do you think about chicken when you are the good example?
Because I like chicken.
You like chicken, you don't like yourself, ne? Hi, boy.
I don't.
He likes chicken more than himself. Alright, so our Trisha or
They are hopeless. Helpless.
They are born helpless.
Nikita.
Cannot fend themselves.
I'm gonna send this video to your mother. Your mother will hear how many times I've told you to concentrate in the class. However, we say, I'm not a preacher, ne? I'm not a pastor, I'm not a bishop, but we say, in my religion, innamal a'mal bil khawatim. It means that the job is counted at last. Nikita, you have been messing up.
So you have 1 minute.
So, no. Nah. It's simple. What I need from you is to change this rate so that you focus. Focus. I want you here in the next session. Alone, not with anybody. Alone. And then I speak when I speak with the air. All right. Lastly, what are the advantages of parental care?
What?
Parental care. guys, advantages of parental care.
Advantages,
Yes.
increase offspring, survival.
Guys, I gave those people specific time to be at school.
That question was also on the other tip that you gave us about the total thing. What are the
Photos.
Yeah.
Tortoise.
Yeah.
So what are advantages of parental care?
Higher chances
Higher
of
chances
survival.
Higher chances of
Survival.
survival. I asked you a question. Remember last time when I asked you a question? You are holding your baby, you go on the street, you find a guy, slaps your baby. What's gonna happen?
Take the baby. Remember, I said he's gonna take the baby.
You're gonna slap me back. I didn't slap you. The guy didn't slap you, slap the baby. So you try to protect the what? The baby. So that the baby can what?
Why was he
Can
slapping?
Yes.
I think they should leave the guy.
You must stop the pain.
She's lying because she doesn't know the pain of pushing.
I won't.
She doesn't know the pain of pushing. All right. Lastly. Lastly. Faith. Tell me 2 things you have learned today. I'm not asking you to look into them. I'm listening. Can I talk about characteristics of dancer?
No.
Rendani, tell me what you have learned today. Guys, I have my babe. Yeah? I have my wife there. Not my wife. My beautiful wife.
Beautiful.
My kids are there. They need daddy. But I have other grown up babies.
We are here.
Now they want to embarrass me. Please don't embarrass me.
Parents must keep their children. Here we won't embarrass you.
Please. Rendani. You know the deal we have, me and you and your daddy? Yeah. Tell me. Tell me what you have learned today. DNA. How about DNA? Guys, I'm coming to you.
DNA is replication that it makes there is a process where DNA makes exact copies of itself, and where does it take place in the interface? Meiosis.
Sean man. Are you okay? I'm just asking if you are okay. Guys, what's the time? What's the time?
It's 9:30.
Half past?
Yes.
20 past.
32 minutes past.
20 past. Is it? What's the time? Okay. Guys, don't cheat. Don't. It's 18 minutes past.
We are finding you.
No, everything was done. Don't always work on time. Round off, ne? Round off.
Yes, yes,
The nearest 10. Not 20.
Even a 5.
Not 20.
It's a 0. 1 0 is a 10.
Alright guys, we are going. It's 20 past. 20 past. No, it's 19. 20 past 9. We are going to break for. It's supposed to be 15 minutes. But listen carefully.
Yeah.
You disturb me, we break for 15 minutes. You don't disturb me, we break for 30 minutes. Make sure that you drink coffee. When you come back, I'm gonna reshuffle you.
But this is fine.
Yeah, so guys, listen. Maximum behavior. I don't want anybody in the class.
Don't worry.
I don't want anybody. I'm coming. Let me fast. Someone was asking questions.